In this week's module, we will talk about normalization for high-throughput genomic data. As a reminder, this is the general flow of a high-throughput genomic experiment. Once an array has been hybridized, there are various pre-processing steps, including image analysis for microarrays and alignment for RNA sequencing assays. This week, we will discuss normalization. What is ultimately of interest in the use of gene expression microarrays is the measurement of differences between experimental and reference states, or between different groups of experimental units. Observed differences in microarray gene expression studies, however, are recognized as arising from two sources of variation, namely, interesting variation in signal intensity and obscuring variation in signal intensity. Interesting variation refers to those differences arising from biological differences between the states being measured. Obscuring or nuisance variation can arise from a host of factors and are of no intrinsic interest. For example, sample handling, the microarray fabrication process, sample processing, processing differences, which could include ambient air humidity that differs by day and by time of year, imbalance in the total amount of RNA from each sample hybridized may differ due to minor pipetting differences. In the case of custom spotted arrays, which uses two labels, differences in dye uptakes, and personnel differences are all examples of obscuring variation, which are not of intrinsic interest. We will use normalization to mitigate these effects so that what we are left with is interesting variation. The purpose of normalization is data correction, that is, altering the observed data to correct for systematic experimental error. Normalization procedures often include removal of variation due to differences in dye affinity effects, spatial location, sample preparation, array differences, and scanning differences. After normalization, all genes on the array that in reality are not differentially expressed should have similar values across all arrays. Note, normalization is not applied to ensure the resulting gene expression data follow a Gaussian or normal distribution. Again, normalization is simply applied to remove sources of obscuring variation. There are two decisions the analyst must make when normalizing a set of microarrays. First, which genes should be used to establish the normalization relationship? Second, what normalization method is most well suited for the given data set? With respect to the first decision, which genes should be used to establish the normalization relationship, one usually decides between housekeeping genes, control genes, an invariant set of genes, or simply you use all genes. With respect to housekeeping genes, those are genes involved in the essential activities of cell maintenance and survival, but not involved in cell function or proliferation. Such genes will be similarly expressed in all samples. One problem in using housekeeping genes is that because they are ubiquitously expressed, they tend to be expressed at high levels. So identifying housekeeping genes that take on the full range of intensities for establishing a normalization relation for all genes on the array is challenging. Control genes are when the experimenter spikes in genes from another organism into the sample at known levels, and these control genes then serve as an artificial housekeeping gene set. They should have equal expression across the arrays. Some problems with using control genes are, first, spots or probes must be included in the assay for any control gene that's spiked into the sample. Second, the experimenter must be very precise with respect to spiking in known amounts of the control genes, which can be technically challenging. With respect to an invariant set of genes, that means that after all arrays have been hybridized, the analyst identifies genes that are relatively constantly expressed across all samples. So these invariant set of genes are empirically determined and will differ from experiment to experiment. Usually, invariant set of genes have expression values in the center of the distribution, and so do not reflect the range of all genes expressed in the samples. All genes may be used, and this is appropriate when the majority of genes are believed to be not differentially expressed across samples. Let's first discuss normalization for custom spotted arrays. The need for normalization has been clearly demonstrated in the custom spotted array setting by examining expression from self-self hybridizations. A self-self hybridization is one in which the same sample is aliquoted into equal-sized subsamples. Each subsample is labeled with one of the fluorescent dyes, and then the two subsamples are pooled and hybridized to the same slide. Since both samples are from the same source, any observed differences should be due to systematic dye bias. Probes are fragments of cDNA spotted in circles by a series of print tips on the surface of the array. However, there are often variations in the amount of deposited probe DNA among spots on the same array. This is often called between print tip variability, but there also may be variability in the amount of probe DNA printed between spots on different arrays for the same print tip, that is, within print tip variability. There also could be differences in the size and shape of deposited target. Additionally, two samples, namely an experimental and reference sample, are labeled and hybridized to one chip. Differences in the labeling process, either due to unequal incorporation of the dye labels, unequal amounts of experimental and reference sample hybridized, or differences in the labeling efficiencies can also be a source of signal variability. A scatter plot is a plot of the values of a random variable y against the values of a second random variable x. Often, when a scatter plot is produced, one examines the resulting plot to assess what type of relationship exists between the two variables. For example, is it linear? In this figure, there is a scatter plot of the red and the green channel intensities from a self-self hybridization. Although we would like for these points to fall along a straight line, clearly they do not. The normalized signal intensity ratio for spot J on array K is given by this equation, where R sub jk denotes the red signal intensity of spot J on array K, G sub jk denotes the green signal of spot J on array K, and C sub jk is the normalization factor for spot J on array K. There are various approaches for calculating C sub jk for custom spotted arrays. This includes global normalization and intensity-dependent normalization. There are various intensity-dependent normalization methods, including loess, invariant set, and quantile normalization. We will look at each in turn.